Welcome to the class of drug discovery. Dear students, on the last turn we have discussed about the introduction of the protein structure, proteins and protein structure. So our today's topic is the protein structure. Okay, in the last class we have discussed about the different amino acids, 20 amino acids, acidic, basic, polar and non-polar amino acids. And we have also discussed about the bonding and interactions, hydrogen bonds and different interactions that are involved in the protein structure. So today we are going to discuss about the structures of the proteins and what are the shapes and sizes of the proteins. So here in this figure you can see these are the so there are the more varieties of the proteins in the cells than any other molecules. So on the basis of shape and sizes we can divide the protein filamentous and globular, large and small. Okay, in the figure you can see these are the various examples of proteins. Phosphor carrier proteins, okay, lysozyme, collagen. You can see this is the fibrous structure. Okay, lysozyme, myoglobin, catalase. So you can see these are the globular structure of protein. Hemoglobin. So this also shows the globular structure because the hemoglobin consists of 4 subunits. Okay, all are globular subunit. Now some protein shows the helical structures also, that is the primary structure or secondary structure of the protein. Deoxyribose nuclease, okay, calmodulin. So these are the various examples: cytochrome C, chymotrypsin, insulin, alcohol dehydrogenase. Some proteins show the triangular structures, but this is a globular structure of proteins. So here in this example, collagen protein is the example of fibrous structure. Okay, now we are going to discuss about secondary structure of protein. What is secondary structure? This can be defined as the localized folding of domains of polypeptide chain. But I will not include domain in the secondary structure. Okay, so because domain is the well-defined subunit of protein that will come in the tertiary structure of protein. Now what is secondary structure? Secondary structure is the some folding of the primary structure. What is primary? Linear chain of amino acid. Now there is a long chain of polypeptide, okay, that is formed, synthesized after transcription of a gene. Now what will be done, what will happen to this long polypeptide chain? Okay, the length of long polypeptide chain is bigger than the cell structure. Now how it can be placed in the small cell structure? Okay, because that is a long polypeptide chain. So in the nature there is a folding of that chain. Okay, so automatically it gets folds. Now how it happens? It depends on the nature of the amino acid, because there is a long chain of amino acids. So between the different amino acid, hydrogen bonds are formed. But for the hydrogen bond formation, atoms should come close together. Then only they will be interacted with each other. So there should be attraction between the atoms. Then the hydrogen bond will be formed. So before the hydrogen bond formation, there are certain interactions we have discussed on the last turn also. These are two types of interaction: van der Waals interactions and electrostatic interaction. These interactions play an important role in the hydrogen bond formation. Without these interactions, hydrogen bond formation do not occur. So van der Waals interaction, if the atoms comes close together, okay, if they cross, they should be close like 1.7 angstrom distance for the hydrogen bond. So if they cross this distance, there will be the formation of hydrogen bonds. So now in the linear chain of polypeptide, if the first amino acid come close to the fourth amino acid, so there will be the formation of hydrogen bond between, because each amino acid contain two terminus, NH2 terminus and carboxyl group, NH2 group and carboxyl group. NH2 group is the amino group, that is it has basic nature. Carboxyl group has acidic nature. So NH2 group of one amino acids make bond with carboxyl group of another atom. So there is the formation of peptide bond. So in that way these different amino acids are connected with each other using peptide bond. In the last lecture we have discussed about that. So due to the formation of hydrogen bond, means first amino acid with fourth amino acids, protein gets helical structure. Now if there is a rope, you can think, so there is a formation of bonds between the first and fourth. So in that way it will be helical structure. Okay. Now how it occurs? This is the linear chain of amino acid. Ramachandran told about the torsion of the torsion angles. What is this torsion angle? If there is a rope and we are stretching this means too much, so there will be the the chain will be strained and restrained. But if we do it in the continuation more frequently, that will be restrained. So there will be the torsion in that chain. In the rope you can see there is a torsion. So due to that it will make the helical structure. So there are certain combination of the angles that is responsible for the helix forms, that is responsible for the protein folding. The protein will get alpha helical structure or protein will get the beta sheet structure. So due to the torsion in the chain, so there is the formation of helix and beta sheets and different types of structure, beta barrels, coils. So okay, now we will look for the different examples of helical structure and beta sheets. So beta sheets is formed by the alpha helix. Okay, this is the again folding of the alpha helical structures. Now in the structure you can see these are the different examples. Amino acid side chain, okay. R group, oxygen group, it is shown in the red color. Hydrogen bonds, okay, there is a hydrogen bond between the two atoms. So you can see it is shown in the light color like red dot. You can see nitrogen, it is shown in the blue color. In the figure B you are able to see between each helical structure there is a distance of 0.54 nanometer. Okay, you can see also the carbon atoms. Again in the figure C you can see the helical structure. So it is okay. Now there is a folding of the different helical structure. So they get fold with each other, they make the different helical structure, make the hydrogen bond with each other, resulting in the formation of beta plated sheet. Here you can see how the hydrogen bond occur between the different helical structures, that is responsible for the formation of beta plated sheets. And there are the two types of hydrogen bond, intermolecular and intramolecular. So these are the between different chains also, different polypeptide, between the polypeptide chain when it gets fold. So there is a turn in the polypeptide chain also. Why turn occur? Because of presence of the proline amino acid. So there are the different amino acids that are present in the polypeptide chain. It depends on the nature of the amino acid. If the proline occur in the linear chain, that means there is a turn in the helix. So turn is responsible for the formation of sheets. And in the binding region, glycine amino acid is present, because glycine is the small amino acid that can only fit it in the small size. It's a protein polypeptide chain in the band region. Okay. Here in this figure you can see these are the hydrophobic amino acids side chains, and there is a hydrogen bond formation also. So it is shown by the red lines. In the figure you can see, and these are the round bowl structure. It shows hydrophobic side chain. Okay. In the figure you can also see the phospholipid. The helix is fundamental in allowing proteins to sit within the plasma membrane. So this is the structure of the membrane. It is the foundation of many very important proteins, visual pigments, immune cells and receptors. So it is clear. So it has been taken from the book Essential Cell Biology. Okay. In the next figure you can see these are the 2,6 alpha helices are coiled together like the strands of a rope. Okay. So this is the helical structure strip of hydrophobic A and D amino acids. Okay. So in the figure you can see these are the amino groups of the helical structure, and these are the carboxyl group of the structures. This is the coil structure. In the figure you can see these are the parallel and anti-parallel sheets. Okay, what is sheet? Sheets means different helical structures come together using the hydrogen bonds. Individual protein domains may ungenerally do consist of combination of secondary structures. So in the figure you can see single helical structure, alpha helix, beta sheets. Okay, and combination of alpha helix and beta pleated sheet and extended regions in which we do not have the helical structure or beta pleated sheet. So that is the second. In the second figure, in the middle of the figure you can see the middle structure shows the combination of all like alpha structures and alpha helical structure and sheet structures and open extended regions which contain the active site also. We will discuss it about this. So this is domain structure. Generally the well defined domain structure is the folding of polypeptide chain in the space. Okay. So protein molecules made of two different domains. So in the third figure you can see the protein consists of two different types of domains. So each domain consists of the helical structures and beta pleated sheets. So in the green color you can see these are the helical structures, and in the red color you can see these are the beta pleated sheets. These are the more figures you can see. First one is an example of pure helical structure with the extended region. In the second figure you can see this is the example of good combination of beta pleated sheet and helical structure. So at the middle you can see how beautifully the beta pleated sheets are arranged. Okay, like the six are shown as the arrow, 6 beta pleated sheet in the above, then above the axis. Okay, and 6 are below the axis. So this is the beautiful combination of the helical and beta pleated sheets. Okay, so only the beta pleated sheets are found at the core region, and in the outer structure there are the helical structure. Okay, in the third structure you can see the third one example is purely consists of the beta pleated sheets and extended regions. Okay, so these are the different examples. Okay, first one is the helical, second one is the helix and beta pleated sheet, and third one is the beta sheets. Nature is known for reusing structure. Proteins are no exception. So here are few examples built on common theme. So but these proteins perform different functions. The The figure, oh okay, that is low. In the figure you can see these are the examples of two different proteins, elastase and chymotrypsin. So these are the two different structures. So you can see there is few difference, but these structures perform different functions. Okay. Although it shows the similarity in the structure, but these structures perform different functions. And these are the other examples, dimer of the cap proteins, in which we have the different domains, and in the second, this is tetramer of neuraminidase proteins. So in the first one structure on the left side, this protein consists of the two domains, dimer. This is dimeric protein, and in the second structure you can see this protein consists of 4 domain. This is a tetramer of the neuraminidase proteins. This is the structure of hemoglobin, which consists of 4 domains. You can see beta domains and alpha domains. So here we see the use of two different polypeptide made by different genes. Hemoglobin is the iron containing oxygen transport metalloprotein in the red blood cells of the bloods in the vertebrates and other animals. So hemoglobin, all of you know, we all contain the hemoglobin protein, which is very essential. Okay, for the respiration also, because it carries the oxygen. So this is all about the protein structures. Students, today we have discussed about the protein structures, shape and sizes of the proteins. We have discussed about the few examples of the proteins. Okay, and we have discussed about the domains also and secondary structure. So these are the filamentous and globular structure. We have seen the examples. These are the different examples. Lysozyme collagen. Collagen is the example of fibrous protein. Catalase is the example of globular protein, and hemoglobin is also the example of globular protein. So these are the different structures. Beta plated sheets are formed by the hydrogen bonding between the helical structures. So these are the examples. Okay, these are the different examples. Okay, and this hemoglobin consists of the 4 domains, which contain the beta domain, 2 beta domain and 2 alpha domain. Okay, and hemoglobin, okay, it works in the oxygen transport systems in the red blood cells of the blood in the vertebrates and other animals. So you know the significance of the hemoglobin. Now lecture is over. Thank you.